I spent the last 40 years of my career as a surgeon at the University of Wisconsin, taking care of diabetic patients. The majority of my patients had no kidney function, so they were in total kidney failure. About 90% had diminished eyesight or were blind, and about 30% already had amputations of various degrees. In an attempt to help these patients, I performed combined kidney pancreas transplants to alleviate their kidney failure, but at the same time free them from insulin injections and hopefully mitigate the secondary complications of type 1 diabetes, which are horrendous and span over a long, long time period, sometimes 20 to 30 years. Now, towards the middle of my efforts, about 20 years after transplanting about 600 of these patients, at the end of my career, I had transplanted about 1,200, and I think I'm still the world record holder as far as this procedure is concerned. But towards the middle of my career, I became terribly frustrated because I was able to help in a good year, as long as organ donors were available, to help perhaps 50 or 60 patients, while at the same time, 40,000 new diabetics were diagnosed in the United States. This did not make any sense to me. So our laboratory, which was primarily engaged in islet transplantation, fetal pancreas transplantation, growth factors, made the decision 20 years ago that gene therapy is most likely the method which will ultimately lead to a cure for diabetes. And this was the reason that towards my retirement, I decided to found a company, and within a very short time, I was extremely fortunate to assemble a group of people who were helping me to establish this company. A few are shown here. Mr. Eric Spira is spearheading our efforts, but we also have Brian Kasper, the inventor of Zolgensma, the second approved gene therapy in the United States, with us, who is and is giving us valuable technical advice. We have Jim Italian, who was responsible for the regulatory pathway which Zolgensma made through the FDA. And we have a number of individuals with long-term experience in the industry and in science. Now, I do not have to tell you what we call the market size. The current number of type 1 diabetics in the United States ranges around 1.5 million. And of course, there is a enlarging number of type 2 diabetics, estimated to be about 30 million in the United States right now. So there is a large number of patients who could benefit from gene therapy for diabetes. Needless to say, diabetes is the leading cause of kidney failure. More patients than for any other indications are currently on the waiting list for a kidney transplant, as most of them are diabetics. It's the leading cause of blindness and the leading cause of non-traumatic amputation. So it is a significant disease, and the healthcare expenses right now are approaching $400 billion per year. Now, 20 years ago, when we made this decision in our laboratory, what route to go, we set ourselves 4 goals. We wanted to be sure that our methodologist, which we develop, is in principle, not right now, but in principle at a later time, available for all diabetic patients, which is in contrast to pancreas transplantation, islet transplantation, even stem cells. Every diabetic patient should have an opportunity to benefit from the therapy. I did not want, despite the fact that I am a surgeon, any surgical intervention, even smaller intervention like implant devices and so on and so forth. In my mind, I had this 7-year-old girl who just developed diabetes. I didn't want to cause this particular girl any trauma at all. All patients receiving a transplant require immunosuppressive therapy, and transplant surgeons know that the immunosuppressive therapy sometimes can do more damage than the actual disease. So no immunosuppression would be allowed. And of course, we have to prove, and we wanted to achieve that with our approach, long-term durability, if possible, with a single treatment. We knew right from the beginning that the Achilles tendon of our approach would be the accurate and precise regulation of glucose metabolism. In particular, we needed to develop methods which avoid the development of low glucose, hypoglycemia, which is the most feared complication which every diabetic has, usually as a result of overdosing of insulin, but sometimes occurring because of nutritional mistakes or other reasons, and is particularly feared in children and keeps mothers alive all night long. So we developed a construct which contained a regulatory element, which we call GIRE, shown in green here, a glucose inducible regulatory element. Now, GIRE will respond to glucose concentrations. If you have a meal and the glucose concentration in the blood rises, GIRE will act as a promoter for the insulin gene. The insulin gene will be expressed, glucose levels will get lower, and GIRE will silence down. So this was about a 5-year effort to get the precise composition of GIRE, and I will show you in a few minutes the results of these efforts. Before I do that, I want to emphasize that our approach is systemic injection of the vector into a peripheral vein with a liver-specific promoter, so the expression of insulin will take place in the hepatocyte. Now, the hepatocyte gives us 2 advantages right from the beginning, perhaps 3. First of all, the hepatocyte is very good in producing peptides or proteins. Second, the hepatocyte already has 2 regulatory elements which come with the cell in its development. One, it has GLUT2, which is a membrane receptor which opens up when the glucose concentration in the blood rises and allows glucose to enter the hepatocyte. And second, glucokinase, which will convert glucose into glycogen. So these are the 2 natural regulatory elements, and we add to that our construct using adeno-associated virus 8, and we're hoping that this construct in combination with hepatocellular therapy will lead to perfect regulation. Before we got to this point, we had to demonstrate that there was no inflammation using our approach. And you can see on the hematoxylin eosin staining that we have no inflammation in the liver. There is no cellular infiltrate. And this is a representative slide of more than 500 rodent experiments. Also, because the hepatocyte does not contain, or the hepatocyte membrane does not contain the autoimmune specific proteins on the surface, the diabetes-related autoimmune related proteins, the hepatocyte is not susceptible to the specific diabetes autoimmune responses. And of course, no immunosuppressive therapy is required. Important to mention is that only 2% of hepatocytes need to be transduced in order to cure a diabetic animal from diabetes. As you can see here on the magnification, the brown cells are cells which have been successfully transduced with our construct, and next to it, an enlarged slide where we estimate that about 1 in 50 cells need to be transduced for sufficient insulin production. Now, how good is our control? The best way to measure control in a diabetic patient or in a diabetic animal is to perform a glucose tolerance test. For the non-clinicians in the audience, a glucose tolerance test in principle provides the animal or the patient with a large amount of glucose at one time within a very short time interval. As a result, and the glucose will rise, and you see on the right slide, you see in a normal animal that, as expected, the glucose concentration will rise to a significant level of about 400 or 500, which of course is 4 or 5 times as high as normal. But then, as expected in a normal animal, within 45, 60, 90 minutes, the glucose values will return to a normal value of about 80 to 100. This would be the normal situation. On the left side, the lower bars are glucose values from animals which were made diabetic and later being treated with our construct. And you can see that these curves are absolutely identical. So the glucose control which we have achieved in our animal by a one-time treatment with adenovirus containing our construct is, one can say, near perfect to a normal, natural situation. On the upper side of the slide is a buffer control, which you see doesn't do anything. So what Endsulin has accomplished in a relatively short time, and I say in a short time because the company is very new, but we are in fact having a longer history than it comes across from this presentation. The laboratory has been working on this concept for about 20 years, and we had a rather seamless transition from the University of Wisconsin laboratory to Endsulin. Basically, within 2 or 3 months, we were out of the starting blocks and were able to conduct experiments. So we are technically a new company, but a lot of the know-how, a lot of what we have done in the laboratory at the University of Wisconsin has been transferred to Endsulin, and we have progressed at a very rapid pace. We have shown that GIRE regulates insulin expression at a near normal level. We have not seen any immune response, neither auto nor allo, and the durability as measured in some of the animals, not all of them, was for the lifetime of the animals, which in some cases went to 18 months, which was the normal lifespan of the animals. At a later time, when we realized that after 90 to 100 days, once we had a straight curve, nothing will change in the long run, we terminated the experiments about 100 days for histology and molecular analysis. So our progress will be testing our lead candidate over the next months and complete preclinical testing within 12 to 18 months, and then we hopefully are on our way to the first clinical trials. Thank you very much for listening.